Polymerase chain reaction. It is discovered by the Kary-Mullis in 1985. It is carried out in vitro, so it is also known or referred as in vitro gene cloning. By using this PCR, we get number of billion of copy of the particular DNA fragment. So if we having one copy of DNA and we want a billion copy, so within a few hours we are getting that billion of copy by using PCR technique. Now PCR is required selected DNA molecule for the amplification purpose, and limitation of this PCR is the border region of DNA which is amplified, and ultimately it attached with the primer at the 3' end. So here we require the border region DNA molecules which attached with the 3' end. Now primer attaching is important in the synthesis of DNA, and we require the double strand primer for this process. So in cycle 1, 5' to 3' primer is used in double strand. Now next, when we are applying the higher temperature, the denaturation of that primer, it means the detachment of that primer is done, and next is annealing. So we are adding or we are attaching the 3' end primer, and ultimately we get the number of copies of selected DNA fragment. This PCR reaction takes place in the Eppendorf tube. So in this Eppendorf tube, we are adding all the 8 substances which are mentioned in the main requirement of PCR. The technique is quick, simple and extremely accurate, so it is used by various scientists in various area of research and diagnosis. Limitation of this PCR is high sensitivity and inaccuracy result when it is get contaminated DNA fragments. Now main requirement of PCR is 2 nucleotide primer complementary to 3' end of target DNA strand. Next, target DNA sequence to produce desired DNA sequence. Third is heat stable DNA polymerase. So this enzyme, this DNA polymerase enzyme is heat stable. So when we are applying higher temperature in denaturation step, it can stand with the heat. So this Taq polymerase is isolated from the bacteria Thermus aquaticus. Next is deoxyadenosine triphosphate, deoxythymide triphosphate, deoxycytidine triphosphate, deoxyglucosine triphosphate, and at the last, all these are introduced into the thermal cycler, and this PCR reaction is carried out in the thermal cycler. The name is suggesting thermal cycler, which provide the thermal, it means heat. Now step involved in PCR is the target DNA sequence, excess of primer, DATP, DTTP, all this ingredient mixed together in the Eppendorf tube, and tube is placed into the thermal cycler. Now reaction mixture is provided with the high temperature, and the temperature range is 90 to 98 degree for the few seconds to denature the DNA. Now consequently the double strand DNA becomes single strand. So here this is happening after denaturation. Next is the temperature is lowered at 55 degree for 20 seconds, so the primer are annealed at 3' end of DNA. So there are 3 steps of PCR reaction. First one is denaturing, second one is annealing, and third one is extension. So in After giving the highest temperature, we are lowering the temperature for some seconds to attach that primer with the DNA end. Now temperature is now maintained at 72 for 30 seconds to facilitate the function of Taq polymerase and to synthesize the complementary strand of the DNA. So we are again increasing the temperature for some period of second, and uh, the Taq polymer give its function in this synthesis. Fifth step, one cycle of the PCR complete, resulting in the formation of two double strand DNA molecule from one double strand DNA. Now, the one cycle of PCR give the two double strand DNA copies. The same cycle is repeated to obtain required number of DNA copies, and the cycle is repeated for the hours, and we get the billion trillion of copies of the DNA. Now, use of PCR. So, for the DNA amplification to detect the mutation process, our gene is mutated, and mutation is the process when our gene is having some unwanted sequence of amino acid in our DNA. So to detect the mutation, it is the kind of vikruti. uh, Next use is for the diagnostic purpose of genetic disorder, for producing in vitro mutation, for preparing DNA for the sequencing. Also, PCR is used for analyzing genetic defects in the single cell from human embryos. Also, it is used to identify the virus and bacteria in the infectious disease. It is also used in the characterization of genotypes. So, this is all done for PCR.